Hello, I'm going to be reviewing the process of oogenesis. This is where diploid germ cells are formed into haploid gametes, and in females, the diploid germ cells are known as oogonium, and these are formed inside the female before she's even born. So before she's even born, she will have created all the oogoniums that she will need throughout her whole life. Millions end up degenerating, and so she's only left with a fraction of what she started off with. The ones that do end up surviving end up going through meiosis 1. And so when this occurs, DNA replication happens and the crossing over genetic material. However, meiosis 1 is paused at prophase 1. So at this stage, the oogonium is now known as a primary oocyte, where it remains diploid. Now, nothing occurs at this point until menstruation. During menstruation, the primary oocyte continues through meiosis 1 and forms a secondary oocyte and a polar body. The reason why this cell is small in comparison to the secondary oocyte is because the cytoplasm is donated. So the polar body ends up degenerating while the secondary oocyte pauses at metaphase 2 of meiosis 2. At this point, the secondary oocyte is haploid. Now, during menstruation, the follicle grows around the secondary oocyte and continues to grow until it reaches a mature follicle. During ovulation, the secondary oocyte with the mature follicle ends up being released into the fallopian tube. Now, during fertilization, a sperm ends up penetrating a secondary oocyte, which resumes meiosis 2. This forms an ootid and a polar body. Now, the polar body, once again, donates its cytoplasm, and so this ends up degenerating while the ootid forms into a mature ovum. Now, the combining of the genetic material of the sperm and the secondary oocyte ends up becoming diploid, and so now forms a zygote.